In this video, we're going to have a look at the cellular immune response. Now, this is really the land of T cells and B cells and what they do. So let's start by talking about T cells and really what are T cells doing for us? They're the ones that are looking at cells that are infected with viruses, bacteria, cancerous cells, abnormal cells, or transplanted cells. Now, in the world of T cells, we're going to see there's many different types. T helper cells, T memory cells, cytotoxic T cells. So some of these cells will go ahead and directly kill cells, and others will release chemicals to mediate the immune response. When we start to look at T cells, we're going to realize quickly they're more complex. So there's more of them, and they have different range of jobs over a B cell. Well, there's two different main populations of T cells we're going to look at, and it's based on their glycoprotein receptors that they have on the outside. One group is going to be called CD4. CD4 will usually be turned into or become activated as helper T cells. And what's the job of these? Well, quickly they can activate B cells, other types of T cells, macrophages, and they have a role in other parts of the immune system, and we'll have a look at that. Some of them also become this group of cells we call reg T cells or regulatory T cells, which moderate the immune response. And a group can also become part of what we'll call memory T cells for use down the road. So CD4 cells can either become T helper cells, T reg, T memory. Those are the main types we're going to be looking at. Now there's another group of T cells. So that was the CD4 we just looked at. There's another whole population which we're going to call CD8, and that's based on what's on the outside of their cell. And they're the ones that will become cytotoxic T cells. And cytotoxic T cells have the ability to recognize virally infected cells or cancer cells and can destroy them directly. Cytotoxic T cells can't go in and fix cells. All they can do is, hey, something's wrong with you, and you will be removed from the cell population. Some of them can also go on and become memory T cells. We're going to see this term. If you're naive, what does this mean as a T cell? This means you've been trained to do your job, but you have not come across the antigen yet that you were specifically trained to find. Until you actually bump into that antigen and become activated, we're going to call you by what's on your cell code. So it's either a CD4 or a CD8. Now once you become activated, then we can start calling you things like a T helper cell or a cytotoxic T cell. And here's just sort of a little sort of a rundown. We're going to go in more details. We make CD4 cells, right? And you notice that they have a given receptor, and we have CD8 cells. Now each one of these goes off and does different jobs. But really, what do we need? We need these cells to basically be activated. They have to find their antigen, and we'll talk about that. And we'll go into more details about how the CD4 cell looks at MHC2 docking sites to see if it can recognize things. If so, it can become activated and proliferate, and we'll talk about those steps in a bit. And same thing over here, CD8 cells. They have a specific T cell receptor, and they're more interested in the class 1 receptors. Now, remember from past discussions is that MHC2 receptors we find on antigen presenting cells. That would be dendritic cells, macrophages, and B cells. That's who that would be. Whereas CD8 cells are eventually, now let me take a second here. Eventually CD8 cells are really concerned about MHC1 proteins and what's being put in them. But we'll get to how these get activated in a bit. The MHC proteins and the antigen, well, really what it is I like to think of this as a little bulletin board. The MHC is a bulletin board. And what it does is shows what is being made in that cell. Let's take an example. Here is an antigen presenting cell. It has found something like a bacteria. It chops it up. It takes a certain segment, like what we call an epitope of it. We place it into the MHC molecule here, which I think is a bulletin board. So it's like I posted something on this bulletin board. And this complex gets moved to the surface to display it, and we'll talk about why it gets displayed. So T cells respond only to processed fragments of antigens that are displayed on the surface of cells in their major histocompatibility complex proteins. And that little chunk that we're going to take from here and put into the bulletin board, we can call it an epitope. And this antigen presentation, why are we doing it? Well, this allows the naive T cells to come up. Now, not every T cell is trained to recognize this shape, but if you're a T cell that recognizes it, then you can turn yourself into what we'll call an effector cell. Basically, you get activated, so you can go on and become a helper T cell, cytotoxic T cell, etc. Now, there are two classes of MHC proteins. There's class 1, which is MHC1s, and MHC2s. Now, where are they? Well, MHC1s we find on all cells except for red blood cells. MHC2s are displayed by antigen-presenting cells, dendritic cells, macrophages, B cells. And these proteins are synthesized in the ER, and what they are, they're that bulletin board that I can basically post stuff on and move it to the cell membrane. So let's talk about MHC1s. MHC1s, it's not a huge thing. It's only like a very small fragment, 8 to 9 amino acids long, and it's an endogenous antigen. Okay? Now think about where MHC1s are. They're on almost every cell except for red blood cell. This is a way for a cell to say what's going on inside itself. So it's going to take endogenous proteins that it's made. If it happens to be, let's say it's a liver cell and it's making normal liver proteins and that, then anything it displays would be a self-antigen. They're normal proteins. But let's say, what if this liver cell was infected by a virus, say hepatitis, one of the hepatitis viruses? Now on that bulletin board is going to be basically non-self antigens, so they're abnormal ones. MHC1s are very critical for activation of CD8s. Now remember, CD8s, when activated, become cytotoxic T cells. Now let's look at the second class, which is the MHC2. We only find these on the antigen presenting cells. Once again, there's a very few cells that are antigen presenting cells. It is, once again, B cells, macrophages, dendritic cells. These proteins, they actually bind with longer fragments. And it's not something made inside the cell. It represents something outside. Because what happens is, let's say this is an APC cell. It finds something foreign like a bacteria. It brings it in, breaks it up, and puts part of it in the MHC2, which gets put on the surface. So what's in here was not made by the APC cell, but it came from the thing it ingested. So it is an exogenous or extracellular. Now, MHC2s are very important for co-stimulation with T helper cells. This is a really good sort of rundown that's in your notes in your book. You can have a look at it. Let's talk about MHC restrictions. So CD4, CD8 cells, they have different requirements for the MHC proteins that present antigens to them. CD4 cells that become things like T helper cells are restricted to binding to only MHC2 proteins, which we only find on APC cells. Now I know these are a lot of initials, so let's take it slowly and we'll have a look at this. So what we're saying is, if you're a CD4 cell, you're only able to dock and interact with cells that have MHC2. And the only cells that have MHC2 are the APC cells, which would be, think about it for a second, macrophages, dendritic cells, and B cells. CD8 cells that become cytotoxic are restricted to binding only to class 1 MHC proteins, which are found on every cell, including APC cells. So this is interesting. APC cells do actually have class 1 also. So APC cells are a weird cell that they have the class 2 that they're going to use to show what's being made inside the cell and show that to T helper cells, but they're also going to have class 1, which is going to allow them to interact with CD8 cells, which become cytotoxic. And we'll come back to that shortly. Once activated, cytotoxic T cells look for the same antigen that they find on class 1 MHC proteins on any cell out there. So let's walk through this. The dilemma for APC cells is how to enable CD8 cells to become activated by non-endogenous phagocytized proteins. One way we can do this is dendritic cells. What they do is they're running around, because let's say if you're a virally infected cell, it's not just one cell. There's millions of the cells are going to be infected, and many of them are going to die as a result of the infection. So this gives a chance for the APC cells to find these dying, dead, infected cells and engulf them. Therefore, now on the, let's say it's a dendritic cell that's engulfed this, now the dendritic cell can put a little bit of what it found on its MHC1 and its MHC2. Because remember, antigen presenting cells are this weird cell that have both types of bulletin boards. So dendrites can have important antigens. Also, this is wild, I should mention this, is that dendritic cells can also form little gap junctions with infected cells rather than engulf them, so they can kind of pull some stuff in through that way, almost like a vacuum cleaner's suction some stuff in that it can then put on its outside surface and show it. That's sort of the start. What we're going to do is we're going to move on, take a break here, start a new video, and talk about the activation and differentiation of T cells. So that would be the follow-up video.